Hi guys, my name is Shad and I'm back teaching the principles of physiology. Today we're going to learn a little bit about muscle. And so let's start off by talking a little bit about some of the pathways that our neurons use to communicate to our muscles. So here I'm drawing the brain is here, that's the spinal cord. That drawing could be a little bit better, but there we go. That's our brain and spinal cord there. So in our brain, we have something called upper motor neurons. And these are a part of a pathway for communicating to our muscles. We also have lower motor neurons, and then we have alpha motor neurons. So I'm going to explain what these things mean here in a second. Let me just get this drawn up. Alright, so here is our nervous system. We have our brain. In our brain we have our upper motor neurons. Down the spinal column we'll have our lower motor neurons. And coming from the spinal column to the muscle fibers is what we call our alpha motor neurons. So we're just going to think about this. This is a concept that we'll talk about a little bit later, but this is just kind of the general pathways of how we communicate to our muscles. We have two types of muscles in our body. We have smooth muscles and we have skeletal muscles. Skeletal muscles are typically voluntary muscles. We can think of something that we want to do and do that action. Me moving my arm like this is controlled by skeletal muscle. It's a voluntary response. Our smooth muscles are typically involuntary. They're just muscles in our body that keep our organs functioning properly and things like that. So as I go throughout this video, these are kind of the pathways that are being activated. So let's jump into some of the things that are actually happening with our muscles. Alright, so in our muscles we have something called the neuromuscular junction. So I'm going to draw that up and then let's explain it right after. So this is one of the neurons that's innervating a muscle. In this neuron up here, we're going to have so if you remember we talked about earlier, we have action potentials. These are things like, this is just basically how our nervous system communicates. But up here on the end of this on the end of this neuron here, we're going to have a calcium voltage gated channel here. So this thing opens up whenever we receive a signal from something down here, typically sodium going into the nerve. So it'll activate this and open this up. So calcium rushes in to this neuron up here. And so calcium goes in here, and that creates what we need to make our neurotransmitters to be released out of this neuron. So as we learned before, we have some snare proteins down here that fuse with our vesicles that contain the neurotransmitters we need to get the response we want. And so here we release this neurotransmitter. Specifically, we're releasing from these neurotransmitters acetylcholine. And down here on our cell, our muscle cell here, we have a couple of receptors. These receptors are nicotinic receptors. We're going to call them the N1 receptors. And acetylcholine binds to these nicotinic receptors. So it'll come down, bind here, bind there, and that will allow sodium to come into the cell. So we bind there, we send a response, sodium comes into the cell, and sodium sends out a signal. We called it the Bwong earlier. The signal gets spread across the cell, and over here we can have other sodium voltage gated channels that are here. They'll be closed, but when the signal reaches them, it can open, it can fire a bunch of other things. We can have a ton of things on here, like our potassium, leak channels. We can have our chlorine channels. There's a ton of things that we can see there. So this whole area is what we call the neuromuscular junction. Over here, we have something that we call a T-tubulin. Can't say that. T-tubulin. In our muscle fibers, we have a lot of these tubulins. They just go deeper into the cell so that we can get a response to different organelles in the cell that we need. And so our T-tubulin goes through, and right next to this T-tubulin, I'm going to draw up another thing. I'm just going to erase some of this. We have our smooth ER here, our sarcoplasmic reticulum, and in this T-tubulin we have we have two parts that come together. Maybe I need to draw this a little better, a little bit closer. But coming from our T-tubulin, we have something called a DHP receptor, which is a dihydropyridine receptor, DHP receptor. It couples with something we call the RYR receptor, which is the rymodine receptor. So RYR receptor. These two things couple together, and it allows for, when we have this sodium that creates a Bwong, it'll reach over here. We'll have a bunch of other voltage gated sodium channels going across the membrane. I can draw another one here. When these things open and allow sodium to come in, it creates that signal that's needed to activate the coupling of these receptors for them to open up. And so they open up, and in our smooth ER, our sarcoplasmic reticulum, we have a ton of calcium buildup in here. Very high amounts of calcium are in here. And so when we have a bunch of calcium building up, we will be able to release that once we get a signal. So the calcium starts coming out of the smooth ER. The way we get it back in is through something that we call the sarcoplasmic endoplasmic calcium ATPase. This is a primary active transport that uses ATP to put calcium back into the smoothie R. And so this is happening within a muscle cell. And these processes are super important because we'll talk about another thing now that is important for muscles. So I'm going to erase this drawing up here. And now we're going to be talking about kind of just the anatomy and structure of what's going on within some of our muscle cells here. So we have something called our Z discs. These are found throughout muscles. There's a ton of them. I'm just going to draw up two right now, but there's typically a lot of these just lying throughout our muscle fibers. And in these Z discs, so this is our Z disc, this straight line up and down. In our Z disc, we have something called nebulin. So nebulin goes across here. And on our nebulin, we have, so let me label that. So the horizontal line is our Z disc. These vertical lines are nebulin. On the nebulin, we can find something that we call actin. So let me express that up here. This is our actin. I'm just going to draw in one. These things are found all over our nebulin. All right, so here, I'll talk about a couple different types of actin, but I'm just going to call this actin for right now. And then on our actin, we have something called tropomyosin, and I'll draw that. So this is our troponin, sorry, that's what we call troponin. We also have tropomyosin on our actin. I'll draw that in a separate picture here in a second, but this is troponin, and let's get a couple more details of this picture and then we can put these pieces together. Oops, sorry, I'm losing my markers. So within, on our Z discs as well, we have something branching out that we call our titin. This is just all throughout as well, coming off of our Z discs there. So this up here, this blue stuff is titin. Coming off of our titin, we have a very important structure, a motor protein, we call it. It is called myosin. So, let me draw that one in green. Oh no, let's do black. So we'll have myosin coming off here. So these black little, I'll draw a better picture of myosin here in a second, but these black lines I'm drawing represent myosin. All right, now let's talk about how all these pieces come together. So, first of all, let's talk about the structure of our actin. And so, actin, we have a couple of parts of it. The single subunits of actin are called G-actin. On our G-actin, we have some binding sites, but I will represent those here in a second. This is our G-actin, and like we said, it's lining our nebulin, right? So I'll draw that nebulin there with G-actin lining it. And then, obviously on both sides. Each of these has a binding site on them. This binding site is specifically for myosin to be able to come. I'll explain that here in a second, but here's our binding sites. For actin, or G-actin. One other component we have here is we have something called tropomyosin. And tropomyosin lines specifically 7 G-actins at a time. So I'll count 1, 2, 3, 4, 5, 6, 7. And it's blocking the active sites of myosin. So this is found in strands of across 7 specific G-actin monomers here. And so when we have this whole system together, we can call this whole thing F-actin. That is F-actin. And now going from that, we have one more thing I want to draw up as a part of this picture. So we have something we call troponin. So this guy right here is our troponin. It's found on both sides as well. Just going to draw one for simplicity. Troponin is super important. Like I said before, this this little blue line that I drew is what we call tropomyosin. And it blocks, specifically binds to 7 sites of our G-actin, and it blocks myosin from being able to bind to actin. So just really quickly while I'm on that topic, let me draw up what a myosin looks like. Looks like this. This is chains that are interwound together, 2 heads. They have, myosin itself has 2 heavy chains and 4 light chains. And myosin, it works to help like I was saying before, it binds to these active sites of G-actin, and it'll actually, if you see here, this myosin's on our titin, and so it helps to like pull Z discs closer together so we can have a muscle contraction. So whenever I contract my muscle like that, myosin is pulling on, just moving across this G-actin here, like walking across it almost, like pulling it. And it pulls these two Z discs closer together, and that's how we can get contractions and relaxation. So moving of those Z discs closer together or further apart. And so the role of troponin is, tropomyosin is blocking our G-actin here. We can't get our myosin to bind to these binding sites on G-actin unless our troponin is out of the way. And so troponin has 3 subunits: an I subunit, a T subunit, and a C subunit. So our I subunit binds to the G-actin. The T subunit binds to our tropomyosin. And our C subunit binds calcium. So we have the T, the I, let's see here. Let's redraw this. C subunit, the T subunit, and the I subunit. This T subunit has 2 binding sites for calcium. It has 2 high affinity and 2 low affinity binding sites. These binding sites are important because when calcium is bound to them, calcium will basically be bound to the high affinity binding sites all the time, but we need calcium bound to the low affinity binding sites as well in order for this mechanism of troponin to work. So what troponin does is when calcium is bound, it moves the tropomyosin out of the way so that we can get our myosin binding to actin. So tropomyosin is kind of like the inhibitor of like contraction, not allowing us to do that. But once we get calcium bound, we can get myosin binding to these active sites, and we can get that contraction to happen. So its job is, once calcium is bound, it removes that tropomyosin. It like moves it out of the way. So it just moves it up and out of the way so that actin can bind to our myosin. All right, so that's that. Now let's talk about just a couple of mechanisms that go behind how myosin works to actually help us with contractions. And so, erase this. Let's start in the next part. So, just quickly before I begin to explain the mechanisms of how the cell works, let me explain kind of some of the sources of ATP that we get in our body and how those sources are used to help us in these mechanisms. So, we have a couple of sources of ATP. Our first one that we use up is our cytosolic ATP. This is just ATP free floating in the cytosol. There's not a lot of it, so it gets used up pretty quick, and it's not really effective for long-term use. This is only used, we can only use this for about 1 to 5 seconds before it's gone, and we can't use it effectively anymore to help us. So it gets used up pretty quickly. and we'll have to go to another source to keep our body going. And so the next source we use is what we call creatine phosphate. And I failed to draw this in the picture before with our Z-disc, so let me just draw a Z-disc out really quick, right? With our nebulin going across here. We have our Titan, right? Within this picture, we have these little enzymes. They're just floating around. We call them creatine phosphate. Not creatine phosphate, my bad, creatine kinase. Sorry about that. Creatine kinase is an enzyme that uses creatine phosphate to make ATP. So creatine kinases have their own source of phosphate they use, and they take ADP and they combine it with the creatine phosphate to make ATP. Now this source of energy is a little bit longer, but it's not that much longer. We get this for about 5 to 10 seconds, and then it's gone. So we have to move to our next source of ATP. That source of ATP is glycolysis, so the breakdown of glucose in our body for energy. And glycolysis, it lasts for about 60 seconds as an energy source. And that is a lot longer, but still, like, if I want to be standing here and talking to you guys for longer than 60 seconds, I need more ATP to keep my body moving. And so these are what we call our anaerobic sources of ATP, because they are found in anaerobic conditions. We have one source in aerobic conditions, and we're going to call this aerobic respiration. This ATP source is found in the mitochondria. And this is basically our electron transport chain. It is taking different reducing agents. It's taking different molecules like FADH2 and NADH and creating ATP from those. And so we get a very much longer source of ATP from this. This can last us up to hours. And so this is going to be the most effective in lasting the longest, while these other ones give us ATP. We use it first, but it doesn't last that long. So we have to keep moving down our sources to make sure that we can keep going. So this is our sources of ATP. Now let's talk about how it relates to the mechanism of myosin binding to our actin. So first, let's talk about, remember, we have two different types of muscles, right? We have skeletal and smooth muscle, but those muscle types also have some subdivisions. Skeletal muscle will only talk about one mechanism. For smooth muscle, there are two mechanisms behind how actin is able to, our myosin's able to bind to our actin. So first off, let's talk about our skeletal muscle. I'm going to draw a cycle and then I'll explain it. It'll be easier if I do that. So here got our Z disk, our nebulin. There is actin on the nebulin, right? And we have myosin right here. And it, oh, that's a bad marker. Let me switch. Let's do green. I'll make this green. So here, ADP. Phosphate. This is what we call the myosin in the cocked phase. So it's cocked right now. We're ready to do something with it. We're ready to get started. We're ready to be able to move along our actin. So myosin's ready to go. It's cocked and loaded. So we have to get that process started. Calcium comes in first. Calcium's going to come in, and it is going to bind to that tropomyosin we were talking about. No, not tropomyosin. It's going to bind to the C subunit of troponin, and when that comes in, it allows our myosin to bind to actin. Now when that happens, we have energy released in two ways. Energy released as heat, and energy used to break the bond that's on phosphate here to our myosin. So that bond gets broken. And so we'll have the phosphate's going to fall off. Eventually we'll have our ADP fall off as well from myosin, and we'll go on to this next phase where we get a power stroke that occurs. So as that power stroke is occurring, we're moving along our actin here. Call this our power stroke. And like I said before, phosphate falls off, and then ADP falls off, and that power stroke happens. In this step, we're going to have ATP rushing in to that binding site in our myosin. And this was cocked. This is what we call the uncocked position here. It's still going to be uncocked, but when we get ATP in here, we have the energy to break away from actin. So myosin breaks away from actin, demonstrated here. We get some energy to do that. Energy used to do that, and we also have energy released as heat. And we can go through hydrolysis to break apart ATP to go back to our first phase. So that happens in skeletal muscle. That's the pathway behind it. Now let's talk about smooth muscle, and we're getting close to the end of this video here. So in our smooth muscle, we have two different cycles that are happening here. We call it the phasic and the tonic cycle. So we'll just compare them side by side. So here's our smooth muscle mechanisms behind how actin is working. So first of all, let's talk about the phasic, and then we'll talk about the tonic. So, similar story here. Actin is always going to start with ADP in its binding site with a phosphate attached. Tonic cycle, same thing. This is in the cocked position, ready to get going, do some action here. First thing that happens in the phasic cycle is calcium comes in to remove that, to get troponin to move the tropomyosin for us. But we have a couple other things that happen. We have another enzyme, MLCK, that is representative of myosin, the myosin light chain kinase. So that comes in as well. And then we'll have ATP coming in as well here. And so the myosin light chain kinase uses a phosphate from ATP and it attaches it onto our model here. So we're still cocked here. We'll just have another ATP bound. So we have not another ATP. We'll have another phosphate bound because the myosin light chain kinase stole that phosphate from ATP and it put it on here. So now, the next step, we're going to have phosphate that falls off because, that bond gets broken, the phosphate falls off, and so will ADP. And here, we are going to have, oh, and I forgot to draw, this is bound up here, right? Same thing, energy released as heat here as that binds on, and then we get energy to break one of these bonds too. So energy breaking a bond to phosphate, energy released as heat. And then in this step of our mechanism, we are going to be in the uncocked position, and phosphate is still bound here. Still the phosphate bound. So one more step here. We have ATP that goes up and in. That ATP is going to bind in the similar site it was bound to before. It's going to be able to remove, actually no, here, my bad. ATP is found bound there still. We're in the cocked position. We still have our phosphate. So the ATP is still bound to myosin here. Still bound to myosin in this picture here, right? We're bound there and we're still attached to actin. And that's because our phosphate here is still attached. So there's two ways we can go here. We can use an enzyme called MLCP, myosin light chain phosphatase, to break off that phosphate, and so the phosphate will leave, and then we undergo hydrolysis again to break apart our ATP into inorganic phosphate and ADP. Or we can go through a cross bridge. This cross bridge phase here allows for us to do another cycle without having to use calcium or ATP or our myosin light chain kinase to get this process moving again, right? So we skip the calcium part here. We skip calcium and we go directly into hydrolyzing the ATP so we can do another round of this. And so that's what we call the phasic. In the phasic, two possible fates when we get to this step. We can either go back and do another round of the cycle, or we can break off that phosphate and start from the beginning again. Two ways we can go. The tonic phase is just a little bit different. So we have the same first step. Calcium comes in, MLCK comes in, and we have ATP coming in. Those strings come in, and we get the same scenario where we have ADP that attaches to our myosin, and then we have phosphates attached, because what's going on up here, we already talked about that, we're in the COX phase, or COX state. But in this step, we have a little bit of different action happening here. We'll have phosphate that falls off because that energy that was released, we have some energy released as heat, energy used to break off that phosphate when we attach our ADP to the myosin, or this, I guess, myosin part gets attached to the actin there as well. So the phosphate falls off, or ADP falls off, same pattern as we've seen before. But the one last thing that's happening here is we have an myosin light chain phosphatase that comes in this step, and it knocks off the other phosphate. And so now we have our power stroke, we're in the uncocked position, but then, so we have no phosphate here. We can't go back to this step over here because we don't have a phosphate. So that's what's different about the tonic cycle, is we can't go back through this cross bridge because we don't have that phosphate. So ATP is going to come in here, and it's going to come in kind of weakly, because we're going to have it in its uncocked phase still, but ATP is going to be trying to bind in there. It'll be falling off. It'll be kind of going back and forth. And this is what creates what we call a latch state. Or this is going to be attached to our myosin for typically, it could be a few minutes, it could be a few hours, but it's going to be attached on longer. It'll be stuck in this uncocked state for longer. And so this is what we call the latch state. ATP is going to be stuck there. Because it can't bind in there efficiently, it's going to take some time to get in there and bind, and once it is bound, we can undergo hydrolysis and go back to our cocked phase and do another round. So that's our tonic versus phasic cycle. Last but not least, let's talk about one more thing. Let's talk about our stretch reflexes versus our, let's just talk about some of the reflexes we have. And the best way I can do this is by describing kind of what's happening. So, I already talked about at the very beginning of this video some of the pathways that our neurons travel in our body to get to our muscle fibers. And we talked about how in our sarcoplasmic reticulum, how an action potential can fire and trigger a series of events of calcium releasing. That calcium is important to bind to troponin, so they can move tropomyosin out of the way, and we can move our muscle fibers together to get a contraction. What I want to talk about now is just some more anatomy terms that we can find in our body. So we have something called, we have bag fibers and chain fibers within a muscle. So I'll draw out a muscle here. All right. And within this muscle, something called bag fiber and a chain fiber. The bag fiber kind of looks like a balloon. The chain fiber just looks like a chain straight up and down. So we have Z discs in here. Nebulin, just lined up and down this thing, right? So that's what I'm drawing here, is just our Z discs with our nebulin. I have some up here, right? And here, this whole thing is what we call the spindle apparatus. This is our spindle apparatus. Within our spindle apparatus, we have intrafusal fibers and extrafusal fibers. So our intrafusal fibers are in here. Within the spindle apparatus, and outside of the spindle apparatus, we have extrafusal fibers. Just outside of the spindle apparatus is our extrafusal fibers. Just some anatomy terms to kind of locate some of the things that we're going to be talking about in our muscles. Let's talk about some differences in the chain fibers and the bag fibers. So this is a bag fiber right here. I'll label it here in a second. And over here we've got our chain fiber. So we label these real quickly. Here, bag fiber. Here's our chain fiber. All right, so in our chain fiber, we find something that we call a 2A muscle fiber. In our bag fibers, we find our 1A fibers. Within our chain fibers, we also find gamma neuron. So we have gamma static motor neurons. And in our bag fibers, we have what we call dynamic gamma motor neurons. So there's our terminology. There's a little bit of the anatomy there. Let's talk about the differences between our bag fibers and our chain fibers. So I'm going to erase that. And we'll get talking here. So here we have a bunch of different stimuluses that can happen in our body. So I'm going to just draw that up. Last but not least. So these are the different types of stimuluses we can experience. Let me all right, so here's our 1A versus our 2A. This is our static. This is our dynamic. So we can call this rate versus duration. And we are in the last part here, so let's talk about some differences here. So, in these stimuluses, before we hit the stimulus, this is called our static area right here, that area can change, right? This is our resting and that's our maintaining. So over here we'll have rest, maintain, static, rest, static, maintain, right? And then we have rest, static, maintain there. We're going to see some differences here. We're going to see some differences of our 1A fibers versus our 2A fibers. Our 2A fibers our 1A fibers are going to be able to contract really fast. You get a lot of stimulus within that amount of time where we're gonna have a lot of stimulation within this static period of time, that period where it's changing. While our duration ones aren't gonna have as many, but they can last longer. So here, let's see a ton of stimulus, while here we'll just see a couple. They're gonna be slower, but they can last longer. So here with our 1A fibers, or type 1A, we are looking at how quickly it's going. We can say we're measuring the rate of how fast our muscle fibers move. Well, this is for how long can they endure to continuously produce a stimulus. And so that's kind of the differences between these guys, is we'll see a lot of stimulus in our 1A fibers, while we're not going to see as much stimulus within our duration. But our duration, this can last very long periods of time. Well, this is just like a quick explosive motion, like for things like sprinting. Well, this would be like the type of muscle fibers a marathon runner might have. And so with our reflexes, we're kind of just like messing with some of that stuff. So our 1A, yeah. So this is our rate, well, that's the duration. And we have different types of muscle fiber in our body. We have both types, but we might have more of one or more of the other. And that would just kind of depend on the type of person we are. And so thank you guys so much for watching this video today. I hope you were able to understand these concepts, and yeah, thanks again for watching. See you later.